Dear friends, welcome to Biology Made Easy. Today we are going to discuss DNA replication part 2. In the previous video in part 1, we have discussed up to certain points how the Y-shaped fork is formed, how they are connected by means of SSG protein so that there is no question of recoiling. So we'll start from that point. And we have discussed up to point number 9. Now we'll discuss point number 10. Now the Y-shaped fork is already formed that we have discussed in the previous video. This is 3 prime, 5 prime, this is 5 prime, and this is 3 prime. Now when DNA replication will take place, both these strands, they can act as a template. But how it will start? The polymerization of the nucleotides will take place by means of DNA polymerase 3. But this DNA polymerase 3 has no capacity to initiate replication because always it requires a free 3' OH group. If this free group is there, then it can attach its phosphate group by forming a ester bond. If there is no, it cannot start. So it has to take the help of another enzyme that is known as the primase. This primase enzyme will synthesize a short strand of RNA primer in this site. A short primer is formed. This is 3 prime, so it will be 5 prime, and this side there will be 3 prime. And in this RNA primer, this is primer, the nucleotides are only of those ribonucleotides, means uracil will be there. And now you see in the 3' end, there will be free OH group. So now DNA polymerase enzyme 3 types, 1, 2, 3, but usually DNA polymerase 3 carries out this function because it carries out very efficiently. So now at this 3' free OH group, the deoxyribonucleotides will be attached, forming ester bonds. And now you see at the 3' it is attaching and it is continuing like this in this direction. In 5' to 3' direction it will continue. And how the nucleotides will be attached? Again in the complementary base sequence. Suppose here it is T, then here it will be A. Suppose here it is G, here it is C, here it is A, here it will be T. Like that, just the complementary sequence they will continue. So this strand, which is formed in the 3' to 5' strand, this is continuous, and this is known as the leading strand. Now what will happen in the other side? Replication will take place simultaneously, but it is slightly different than the other side. What is the reason behind it? You see, this DNA template, it runs from 3' to 5', so that the replication takes place in 5' to 3'. Because replication can never take place in the reverse direction, 3' to 5', never, it is not possible. Because DNA polymerase will never catalyze the reactions. So when the Y-shaped fork is formed, in this site, the DNA will be formed only in fragments. Suppose the DNA is slightly open, then one primer will be formed, and then the DNA will be synthesized in this direction. That means you see this is 5', so this is 3', this is 5', that means in the 5 to 3 direction is taking place. Again when the DNA will be slightly separated, again one primer is formed, and again DNA is formed. Again it opened, and again one primer is formed, and again DNA replication takes place. That means in this side, in 5' to 3' that acts as a template, here the new DNA molecules were synthesized, they are formed in fragments Each fragment is known as Okazaki fragment. Again you mark, these are formed discontinuously and in opposite direction to this leading strand. It is taking place in this direction, it is taking place in this direction, but in either case, 5' to 3', 5' to 3'. That will be never opposite, only the direction is opposite, but 5' to 3', that is constant. So, what will be the number of primers? It depends upon the number of Okazaki fragments. And this side, there is a single primer. And in all these primers, there are only ribonucleotides, which are not necessary for DNA. So, the next step, 11th step, removal of primers. Now, these primers which have ribonucleotides, they will be removed. But who will remove it? Which enzyme will remove it? Already I have told you that DNA polymerase 3 types, 1, 2, 3. Now, the removal of these primers is the function of DNA polymerase 1. You may ask here, why the polymerization is taking place by DNA polymerase 3, but while removing, why DNA polymerase 1 is coming? That is very, very important. Because the DNA polymerase, which are 3 types, DNA polymerase 1, 2, 3, all of them have 5' to 3' polymerase. All of them can polymerize in 5' to 3'. Again, all of them have exonuclease activity 3' to 5'. Exonuclease means removal. This is 3' to 5'. They can remove one nucleotides from the 3' end, but not from that side. But you see, when the primer should be removed, the primer should be removed only from the 5', not from the 3'. That means the DNA polymerase, which has capacity of 5' to 3' exonuclease activity, that can only do this function. And that is the function of a DNA polymerase 1. Now, this DNA polymerase 1 having capacity of 5' to 3' active exonuclease activity, it will remove the primers. And again, it will add new nucleotides of DNA according to the complementary sequence. So now you see the primers are removed. Again, in between the Okazaki fragments, still some gaps are there. So those gaps are to be filled. So that is the function of another enzyme, DNA ligase. Now DNA ligase will join all these fragments. So now a continuous strand is formed in the 5' to 3'. Now this is known as lagging strand. So you see, in the 3' to 5' DNA template, the new DNA which is synthesized continuously is known as leading strand. But in the 5' to 3', the DNA which is synthesized again in the 5' to 3' direction but opposite, it is synthesized discontinuously and known as lagging strand. Now you mark one thing, one strand is synthesized continuously, the other strand is synthesized discontinuously. So if somebody will ask us whether the replication is continuous or discontinuous, what will tell? We'll combine to semi-discontinuous. It is semi-discontinuous. That means DNA replication is bidirectional, semi-conservative, and semi-discontinuous. So now the DNA replication on either side and both the templates is completed. But if there will be a single mistake in the DNA, it will cause mutation. And the mutation cannot be tolerated by higher organisms, even in lower organisms. But if in lower organisms there is a mutation and they will die, that will not come as breaking news. But if it will take place in higher organisms, breaking news. So here we have to see how this is corrected. If there is any mistake, how it is corrected. Now that correction is known as proofreading or editing. Next step, proofreading or editing. So now this function is again carried out by DNA polymerase 1. Now DNA polymerase 1 will now inspect the entire length of the newly synthesized DNA if there is any mistake. And if at any point there is any mistake, it will remove it and again it will add the correct one. This is known as proofreading and editing. Now if in the DNA there are so many errors, so many defects are there, and the DNA polymerase cannot correct all of them in the right time, then it will lead to a stage which is known as apoptosis, means programmed death. In that case, the cell is destined to die. Now, the next point. Now, the replication is complete, but where it will be terminated? For termination, there is a particular site known as TOR site, at which a TOS protein is attached. TOR site means terminal site to which a TOS protein is attached, and that point will be recognized by DNA polymerase, and there the replication will stop. Next point. The replication can be inhibited. It can be inhibited by ethidium bromide. If it is given, replication cannot take place. Next point. In cell division, we know that in each step, the telomeric ends, they decrease in size. For each cell division, there is a decrease of 200 base pairs. And during this replication, the depletion of those 200 base pairs are replaced by one enzyme, telomerase. This telomerase is a reverse transcriptase type of enzyme, and it synthesizes the DNA at the telomeric ends, so that again it can divide. Again there will be decrease, again it will divide. That means the telomerase, if the telomerase enzyme is present in the cell will always go on dividing. But if this telomerase enzyme is very active in somatic cells, what will happen? It will lead to cancer. That is why in cancer treatment, doctors try to inhibit the activity of this telomerase enzyme so that cancer can be controlled. Then, in case of prokaryotes like bacteria, in bacteria it is a double-stranded circular DNA. So when there will be replication, the two strands will be separated, and during half of replication, it will give an appearance like this. Means it gives the appearance of a theta. That means in eukaryotes, while the DNA replication gives a Y-shaped fork, in case of a prokaryotes, it gives a theta appearance. So these are some of the very, very important points regarding the DNA replication. I hope you have understood it perfectly. And now I will give you a few questions. Question number 1. Y-shaped fork is formed in which stage of cell cycle? A. G1, B. S, C. G2, and D choice M phase. Second question. Lagging strand is synthesized in which DNA template? A choice 3 prime to 5 prime, B choice 5 prime to 3 prime, C choice possibly in both, D none. The next question. This question is frequently asked in examinations. If the sequence of nucleotides in a DNA template is 5 prime, A, G, C, T, G, A, 3 prime, then what will be the corresponding nucleotides in the other strand? So A choice 5 prime T, C, G, A, C, T, 3 prime. B choice 3 prime, see the same one, T, C, G, A, C, T, 5 prime. C choice 3 prime, A, G, C, T, G, A, 5 prime. Then D choice 5 prime, A, G, T, C, G, A, 3 prime. I hope you will be able to answer all these questions, and again I hope you have understood their application perfectly. So thank you very much for watching it. Have a good day.